Hello everybody. Today I'm going to present about chloroplasts. Let us begin. Okay, evolutionary history for chloroplasts. As you see in the diagram, and you understand that chloroplasts originally derived from swallowed prokaryotes that once existed as distinct species. At some point, an aerobic prokaryote was engulfed by eukaryotic cell, which is you see in here, which can develop an endosymbiotic relationship with the host eukaryote, eventually transforming into a mitochondrion. At the end of this, photosynthetic prokaryotes were first observed by eukaryote cell comprising mitochondria, which developed into specialized chloroplast organelles. So next, let us understand the function and the structure of chloroplasts. So as you see, the picture that I draw, you see lumen, outer membrane, renum, endomembrane, stroma, intermembrane, stroma lamella, thylakoid, and so on. So let us understand the function of this structure. First, the outer membrane. It is a semi-porous membrane and is permeable to small molecules and ions, which diffuse easily. The outer membrane is not permeable to larger protein. Intermembrane space. It is usually a thin intermembrane space about 10 to 20 nanometers, and it is present between the outer and the inner membrane of the chloroplast. Inner membrane. The inner membrane of the chloroplast form a border to the stroma. It regulates the phases of materials in and out of the chloroplast. In addition to regulation activity, fatty acids, lipids, and carotenoids are synthesized in the inner chloroplast membrane. Stroma. Stroma is an alkaline aqueous fluid that is protein-rich and is present within the inner membrane of the chloroplast. The space outside the thylakoid space is called the stroma. The chloroplast DNA, chloroplast ribosomes, and the thylakoid system, starch granules, and many proteins are found floating around the stroma. Lastly, thylakoid system. The thylakoid system is suspended in the stroma. The thylakoid system is a collection of membranous sacs called thylakoids. The chlorophyll is found in the thylakoid and is the site for the process of light reactions of photosynthesis to happen. The thylakoids are arranged in stacks known as grana. Each granum contains around 10 to 20 thylakoids. So, what is the genes that play important roles in the function of chloroplasts? First of all is rRNA genes, which is associated with a set of proteins to form ribosomes. This complex structure, which physically move along an mRNA molecules, catalyze the assembly of amino acids into protein chains. They also bind tRNAs and various accessory molecules necessary for protein synthesis. tRNA genes. Since chloroplasts encode a complete and independent genome, it is important to study the chloroplast genomes, especially chloroplast tRNAs, which are responsible for protein translation. Ribosomal proteins. They are responsible for energy conversion and carbon fixations by photosynthetic reaction in plant and algae. And the reagents, which is Robisco. Robisco is the key enzyme responsible for photosynthetic carbon assimilation in catalyzing the reaction of CO2 with ribulose 1,5 biphosphate to form two molecules of phosphoglyceric acid, which is PGA. So, between that two, the protein coding gene is ribosomal and Robisco. So, why are the two common use genes more commonly used than the other two genes, which is not the ribosomal and Robisco? Because there are four reasons. First, it protein synthesis. Then, to maintain energy flow, to ensure the life of the plant, and for growth of the growth purpose. Because as we know, a plant are independent, so they need to generate their own food. So protein is very important for them. So these two genes are very common in all plant that consist in the world. So before I end my lecture, this is the reference that I use. If you have any question, you can contact me, Muhammad Shakendi. Thank you.